Hello everyone, myself Ms.Namita Garude, Assistant Professor in Microbiology. Let us study Agrose gel electrophoresis. What is electrophoresis? Electrophoresis is a method of separation of charged biological molecules like DNA, RNA and protein under the influence of electric field. The separation takes place on the basis of molecular size and molecular weight. Agrose gel electrophoresis is used for the separation of DNA and RNA. Agarose is a polysaccharide obtained from the red algae Porphyra umbilicalis. It is made up of repetitive subunits of agarobiotes, which comprise alternative units of galactose and 3,6 anhydrogalactose. The equipment to carry out electrophoresis is electrophoresis chamber and power supply. Agrose solution, gel casting tray, which are available in variety of sizes and composed of UV transparent plastic. Plastic comb, which around which molten agarose is poured to form sample well in the gel. The DNA samples and DNA ladders. Electrophoresis buffer, usually tris acetate EDTA or tris borate EDTA used as a buffer. Loading buffer, which contains something dense, for example glycerol, allow the sample to fall into the sample veins, and also contains a tracking dye, for example bromophenol blue, which migrate in the gel and allow visualization how far the electrophoresis has proceeded. Ethidium bromide, a fluorescent dye used to stain the nucleic acid. Transilluminator, required for the visualization of ethidium bromide stain DNA in the gel. The negatively charged DNA will migrate towards the positive charge under the influence of constant current. Thus, the separation depends on the mass and charge of the DNA. The DNA molecules are forced to move through the agarose gel pores. The rate of migration depends on various factors. For example, strength of the electric field, hydrophobicity of the DNA, ionic strength of the buffer, the size and shape of the DNA, temperature of the buffer. Most agarose gels are made between 0.7% to 2% of the agarose. 0.7% gel is suitable for the separation of large DNA fragments up to 10 kb, and 2% gel is suitable for the good resolution for the small fragments with the size of 0.2 to 1 kb. By agarose gel electrophoresis, the smaller fragments of the DNA move faster as compared to the large fragments of the DNA. Applications of the agarose gel electrophoresis are restriction mapping of clone DNA, analysis of PCR products, separation of restricted genomic DNA prior to the southern and northern blotting. Agarose gel electrophoresis is the method of gel electrophoresis used in biochemistry, molecular biology and chemical chemistry to separate a mixture population of DNA or a protein in a mixture of agarose. Biomolecules are separated by applying electric field to move charged molecule through the agarose matrix, and the biomolecules are separated by size in the agarose gel matrix. Thank you.